This presentation gives an overview of the research article, Astrocytes, phagocytose adult hippocampal synapses for circuit homeostasis by Joon Hyuk Lee et al., published in the journal Nature in 2020. When people think about how the brain functions, the first thing they think of are neurons. Not many people tend to think about glial cells or synapses, but glial cells are actually very important. To put it simply, they play a supporting role to neurons and they provide things like nutrients and protection. You can kind of think of them as the people who work behind the scenes for movie productions. They're not the main characters, but without them, you wouldn't be able to produce the movie. So in this diagram, you can see that there's a lot going on, but only two of these, the yellow ones, are neurons. The rest are glial cells. There are many types of glial cells, but the ones we will focus on today are astrocytes, which are the green ones down here. One of their major roles is to manage synapses, which are the space in between neurons through which signals are passed. So here, you can see two neurons that are not quite touching each other. The space in between them is called the synapse. It's important to have synapses that function well and to get rid of synapses that aren't. This is called synaptic pruning. So think of when you're editing an essay. You want to get rid of sentences that don't make sense, and you also want to add new ones that help strengthen your essay. That's basically what synaptic pruning is. Astrocytes act as the executor that gets rid of the synapses by swallowing them, and this process is known as phagocytosis. You can see that in this diagram right here. You can see the astrocyte is swallowing the bacteria up here.
So the researchers were able to identify areas in which synapse elimination occurred. They did this using fluorescence. There is a way to induce two proteins called mCherry and EGFP. These proteins glow red and green respectively when put under light. Both proteins are found at the synapse, and the combination of the colors makes us see yellow when they're together. That's what you see on the right side of this figure. On the left side, we see an area of only red. This is where mCherry exists alone. As we mentioned earlier, astrocytes will destroy synapses by swallowing them. In order to destroy something, acid is required. Only the red protein is able to survive in acidic environments. Green protein stops working. In those areas where you see only red, you know that the green protein had stopped working. This indicates that these proteins are now in an acidic environment, which is indicative of synaptic pruning or editing. If you shine light on some molecules, you may see light of a different color emitted from those molecules. This is known as fluorescence. Think of it as the UV light that detectives use to find hidden fingerprints in crime scenes. The molecules absorb high energy light, for example, blue in this image. This increases the energy of the molecules, which is represented as the top black line. Some of the energy is lost internally, which is the red line. The molecule then emits light with a less energy, green in this example. Fluorescence is a common dye that acts in exactly this way. The advantage of fluorescence for microscopy is that you can often attach fluorescent dye molecules to specific parts of your sample so that only those parts are the ones seen in microscope. You can also use more than one type of dye. By changing the excitation light, you can cause one type of dye to fluoresce and then another to distinguish two different parts of your sample.
In order to look at the specimen while the fluorescent dyed proteins are still active, a light microscopy is research, and it is called confocal microscopy. Confocal microscopy uses a small blocker called pinhole to block autofocus light. This pinhole blocks the light above and below the desired imaging area, which increases the resolution and allows you to view a thick piece of specimen, while a regular light microscope has prepared samples that need to be sliced very thinly in order to be imaged. Otherwise, its resolution would be affected by the background. Here, in this image on the right, we can see that the signals from above and below the specimen are being blocked by the blocker called pinhole. And an example of the confocal microscopy image is shown at the center of the slide. And although a big volume of the sample is being used, only the selected section of it is imaged, and the red and green fluorescence proteins are still visible. As mentioned earlier, the presence of red proteins shows that there was editing. In this figure, you can see the amounts of red protein found in the X-PRIT, which is a type of excitatory synapse, a synapse that induces more activity. When we compare this to the next figure, you can see that X-PRIT and even the X-POST had much greater amounts of the red protein than in the inhibit or the inhipost, which are inhibitory synapses and induce less activity. This shows that the astrocytes probably control editing more for synapses that induce more activity than the ones that don't. In conclusion, the use of fluorescence in confocal microscopy with M-cherry and EGFP proteins helped researchers find that astrocytes contribute to synaptic editing, especially for the most active synapses. This could be very important for people whose brains are damaged and cannot edit their synapses, such as in cases of autism or Alzheimer's, because it shows that astrocytes can perform this crucial function. Thus, it opens up the possibility of using astrocytes as a future treatment.